Am I audible?
Yes.
Okay, so let's do it. Bismillah ar-Rahman ar-Rahim. In the name of Allah, most gracious, most merciful, I would like to start our virtual seminar titled Microalgal Self-Factor-Risk, the fourth generation perspective for industrial biofuels. Assalamu alaikum, good morning, good afternoon, good evening, everyone, depending on wherever you are watching us live on Zoom, Facebook, and YouTube. I'm your host, Assam Shurkar from Bangladesh. Welcome to the seminar. There are three primary ways you can participate in today's event. The first 250 registered people have joined us with the panel via Zoom app. Others are watching us live on Facebook and YouTube. So if you face any difficulties in one of the platforms, then please consider trying another. For Facebook, please visit tiny.cc/SPPseminars_03. Or you can watch us live from SciencePodabD's official Facebook page at fb.com/SciencePodabD. For YouTube, please visit tiny.cc/SPPseminars_02. You can find all the links at SciencePodabD's official Facebook page at fb.com/SciencePodabD. Dear Zoom attendees, please make sure to mute your microphones other than the panel so we can understand our speaker better. And for the panel, please mute your microphones other than the speaker so we do not get any background noises and participants have no difficulty understanding us. Dear attendees, please put on your headphones for the best ever experience. But still, if you face any difficulties whatsoever, please let us know in the comment section. We will try to fix them as soon as possible. First of all, I'd like to thank our partners and sponsors. Virtual seminar on the production of fourth generation biofuels is brought to you by SciencePodabD, sponsored by Cell Genetics and Plant Biotechnology Lab. SciencePodabD is a scientific communication platform based in Bangladesh. Members are highly enthusiastic to explore and do science. We communicate and collaborate with other interested groups and peoples who are striving for a common goal from all around the world. This has been our second international initiative. Our first virtual seminar on genome editing entitled The Basics of Genome Editing, What, Why, and How by Dr. Kidibuddin Alimolla was a huge success. We had 716 registered participants from more than 20 countries, and it was well received by the scientific community as well. You can watch the recorded video at tiny.cc/genome_editing. We will continue to bring such events in the future as well to make the world a much better place for the future generation. So please make sure to subscribe to our YouTube channel and like and follow our official Facebook page at fb.com/SciencePodabD. I would like to thank J Computer Club, J Study Club, JU Biotech Club, and other organizations for allowing us to use their space for our promotion. I'd like to thank our university partners, Jarif Mahbub Sabon from the University of Dhaka, Shamim Afroz from the University of Rajshahi, Abid Muqtadir from the University of Kulna. Without their active efforts, we couldn't reach to you guys. I'd also like to thank our technical team, Nazmin Islam Akash for YouTube streaming, Zanatul Firdaus, Mahfuz Mo, Mahmudul Hassan Rifat for Facebook streaming, first stream from Jahangirnagar University, and Mahmudul Hassan Rifat from Sylhet Agricultural University. I'd like to thank our social media team, Mahfuz Mo, MD Luqman Hussain, Sabir Hussain, and Farhad Hussain, all from Jahangirnagar University for doing an amazing job on social media. And of course, our print media partners for covering us in the print media as well. And finally, I'd like to thank you, the audience. We were overwhelmed by your responses. We have received 453 registrations from all around the world. Without your continued support, we couldn't be here today. It would not be possible without the active contribution of Science for the Bangladesh team. So a big round of applause for you guys as well. Without further ado, let's introduce you to our panel. Let's meet today's event chair, Dr. Abdullah Mohammad Shohail, Professor of Biotechnology and Genetic Engineering at Jahangirnagar University, Bangladesh. Dr. Shohail is a respected authority in the field of biotechnology. But for those of you who don't know him, he is the Director of Cell Genetics and Plant Biotechnology Lab. He did his Ph.D. in Agricultural Biotechnology from Chumpak National University, South Korea. He had worked as a research collaborator for the International Rice Research Institute in the Philippines. He was awarded the prestigious JASPS Postdoctoral Fellowship. He had worked as a Postdoctoral Research Associate at the Cell Genetics and Citrus Improvement Lab at the University of Florida. Dr. Shohail has published articles in many reputed international journals. We are lucky to have one of our alumni, now a lecturer at Bangabandhu Sheikh Mujibur Rahman Agricultural University, Tanbir Ahmed Piash, with us in the panel. And now, the attraction for today's event. We are very delighted to have one of the leading experts in biofuels, Dr. Pavan Jutur from New Delhi, joined with us in the panel. Welcome, Dr. Jutur. Dr. Jutur is a well-known figure. There's no introduction, but for those of you who don't know him, Dr. Jutur is the group leader of Omics of Algae at the International Center for Genetic Engineering and Biotechnology. Dr. Jutur has done his M.Phil and Ph.D. in Biochemistry Molecular Biology from Pondicherry University, India. Then he continued his research as a Postdoctoral Research Associate at the University of Nebraska in the USA till 2012. Later in the same year, he joined ICGEB. His primary focus of research is understanding the dynamics of microalgal systems through integrated multi-omics approach in accordance with reconstructed genome-scale metabolic models and well-defined functional pathways that will elucidate an effective strategy for converting carbon sources to biomass, biofuels, and biorenewables for sustainable futuristic solutions. His findings will possibly provide an important breakthrough on central metabolism of microalgae, which is required for next-generation biofuel production. Enough intro, let's dig in. Let me give you a rough overview of today's event. First, our speaker, Dr. Jutur, will talk about the production of fourth-generation biofuels in great detail. Then today's event chair, Dr. Abdullah Mamashwal, will greet you with his message. Then our alumni, Tanbir Ahmed Piash, will greet you with his message. In the meantime, our team will collect your questions and we will go for a Q&A session shortly. So make sure to leave your questions in the comment sections of Zoom, Facebook, and YouTube Live ahead of time. Dr. Jutur, the floor is all yours.
Oh, thank you very much, Sam, for the introduction. And I would first of all thank Professor Soil for inviting me to give a guest talk on this virtual fourth generation biofuels. And I'm happy and delighted to deliver a talk and also highlight our research perspective, what we are doing at ICGEB. So welcome all audience around the world, especially students, graduates, undergraduate students, postdocs, and faculty members of different universities. And I would like to introduce myself. I'm Pavan Jutur, working as a group leader for Omics of Algae at ICGEB, New Delhi, India. So basically, Sam has introduced me what our research interests. So I'll be going through that in my talk for this hour and a half, and I will be delighted to answer your questions at the later of the section. So I'm happy. And first to start with, so today I'm going to focus my talk on microalgal cell factories, otherwise known as photosynthetic green factories. The fourth generation perspective for industrial biofuels. So all these industrial biofuels have gained much relevance in this last decade, and we have been seeing like great interest in research, industrial sector in every area of biofuel, which has been enhanced in due course of time. So in no delay, I'm just starting with introduction of So in order to start with, let us go ahead with what is ICGEB and where we are located with. ICGEB is known as International Center for Genetic Engineering and Biotechnology, an intergovernmental organization for research, training, technology transfer in life sciences to promote sustainable global development. And we are located in three places primarily. Our headquarters is in Trieste, Italy. I'm currently located in New Delhi here, and there is a third campus which is in South Africa. And we have different member states which are enrolled, which are the members of the ICGEB, and we call it as member states. And all the member states, including Bangladesh, is one of the member states of that. And so we promote research and collaboration with the member states and non-member states. So there are 22 signatory countries involved in that. And as I said, there are 3 host campuses components of ICGEB. So basically, there are 3 major components at ICGEB New Delhi. The one is human health, the second one is plant biology, and the third one is industrial biofuels. So the different groups work on different areas. But the core research comes under these 3 umbrellas. So ICGEB, as you know, that it's very popular for its fellowship program. It's called Arthropolsky Fellowship, which is having pre-doctoral, postdoctoral, short-term, and short-term fellowship, which are very popular and highly competitive. And every year there is a call in March and September for these fellowships, and all the international students are eligible to apply for these fellowships with respect to what research area they are interested in. So this is some statistics from where you can see that a lot of people coming out from different countries participated in ICGEB components, in different components. And every year we have different international students who get graduated from different components of ICGEB. So we have networking with other components, other supporting scientists supported by smart fellowship programs. So that finally, ICGEB is also well known for its meetings and courses, well-defined meetings and courses in different areas of research. And we call it as ICGEB meetings and calling courses. And you can look in the website. And every year we have a series of courses and meeting programs at different locations based on the time and duration. And for scientists and the faculty, we have a CRP collaborative research program, which you can apply for the grants to ICGEB, independent grants, and ICGEB supports good grants in the area of interest, and also in collaboration with the labs of ICGEB. So these are the very, very confirmative and very promising programs which ICGEB conduct on day-to-day basis or yearly basis, and you can check the website for the latest updates. So with this a small introduction to ICGEB, I would like to go forward and carry forward with my talk today. And my talk basically will comprise of these following contents. So first of all, I will be discussing what biofuel generations, about the biofuel generation. So we are talking about first generation, second generation, third generation, and fourth generation. What are these, and why should we be much interested in that? So that the major question is, do we need why we need the production of biofuels, and biofuels are the very important, as I told, why we need the biofuels in that concern. The third one is the classification of algae, and why algae, because we are mostly, our group is interested in looking for algal biofuels, and we'll be discussing more on algal biofuels, and basically the paradigm in algal biotechnology, how we try to learn from these algal consortiums. Finally, omics of algal and amicable solution, what we are looking to understand the algae as a precursor for future biofuel generation. Finally, a small glance at fourth generation biofuels, exactly what we are looking for and how it will be in near future. And this is a promising area of research for especially for undergraduates and graduates and postdoctoral students. And finally, a small example of how an NREL-based research group has come up with a roadmap where they can produce an algal biofuels at $7.5 per gallon. So everything, because there is a big question that is algal biofuels is sustainable or is it promising? So that can be answered through this roadmap. So these are the main outline of my talk today. And if you have any questions, please write me down the questions, and I will try to answer at the end of my talk. Thank you. So first, we should understand what is the scenario and the prediction on global petroleum sources. So we know that in 2006, it was predominantly like conversion petroleum is used as 93.2 million barrels per day, and it is estimated to predict that by 2030 it requires around 106.6 million barrels per day. Maybe it may be a conventional fuels, it may be biofuel, it may be coal to fuel. So all these together, we need this high amount of petroleum sources in order to sustain and in order to do our major challenges from that. So how you can sustain these, because we know that fossil fuels are getting depleted and there should be an alternative. And this is a futuristic research. This is not a research solution for just today. And this is a futuristic research in the coming years. So we have to think ahead for next 50 to 100 years, what will be the alternative for a fossil fuel if fossil fuels are getting diminished and there is a requirement for alternative generation of fuels. So in that scenario, we are talking about biofuel generations. Mainly, most of you guys may have heard about first generation and second generation and third generation biofuels. Basically, I'll just go through each component of this pictorial representation, where you can see that first is raw material selection. That is first generation, second generation, or third or fourth generation. So what kind of raw material you are selecting for a particular this thing comes under this generation of that. And so first generation is mainly from edible crops like sugarcane, rice, wheat, potato, sugar beet, etc. And the first generation biofuels has always had a hurdle, that food versus fuel issue crisis, that you cannot compromise on your food negotiation, because food is also equally, edible crops are also equally important in terms of the daily requirements of the global population. So as an alternative, second generation biofuels from waste and lignocellulosic biomass, such as sugarcane biomass, bagasse, forest residues, grass, cell biomass from fermentation, etc., are the second generation biofuels which are being very predominant. And you can see that there are already a few establishments with second generation biofuels for the production of bioethanol and all. So it's a very popular this thing. And third generation biofuels is especially from algae. So algae does not only produce oils, but also produce high value products. That's why it is essentially another biocommodity where you can look for multiple products simultaneously and the fourth generation is non-edible raw materials such as CO2, jatropha, castor, carrageenan. These are the fourth generation non-edible crops which can be used in the course, and how we will be using this generation. And this is actually more interconnected with the third generation and second generation biofuels, in a context that the improvements of these generation are going to the fourth generation, where you can capture the final my final part will show you how the fourth generation will be projected in the near future. And there are huge areas of research which has to be worked out on the third generation, second generation, and fourth generation biofuels, where it is important for each of us to understand each component and troubleshoot the challenges so that we'll achieve the successful for the futuristic perspectives. The second thing is the technology of transformation of selection, where there's different components which convert the raw material to the final product. So those are mainly four, that is physical may be ultrasound, mechanical extraction. All these things come under physical extraction procedures. The second one is thermochemical, where pyrolysis of the raw material, gasification, liquefaction, and combustion. These are the processes where you convert the basic raw material and extract the sugars or whatever molecules you need to convert into final byproduct. And chemical process like hydrolysis, solvent extraction, supercritical extraction of the chemical process which are involved. And finally, there is one more process called biochemical process, where biocatalyst, anaerobic and aerobic fermentation comes into the role. So the well popular known thing is fermentation is bioethanol by yeast, Saccharomyces cerevisiae, and it's a very popular and well established throughout. And there are many industries which work on that scenario. Finally, what the products we are going to see is biofuels, bioenergy, biomaterials, and biochemicals, and different high value products which are even food products, fish feed, cattle feed, all these things. So biofertilizers, biochar, all these components are the products which are being upturned. So as I said, like all these generation biofuels finally lead to a products of selection, multiple products which can be simultaneously of great significance. But still, there is a lacuna in the research perspective of this, and people are working throughout the world in different areas, even with second generation, third generation, and fourth generation, to make amicable solution from different perspectives of understanding how this can be from basic laboratory to it can go to a translational, that is industrial mode or a commercial mode. So thus biofuel generations are basically characterized into four types of generations, and my research will be oriented in understanding more on third generation biofuel, where we are trying to understand the physiology or basic molecular metabolic biochemical pathways of the algal system, so that it can how we can produce the biomolecules or products of different high value importance. So as I said, why we need the production, as you know, fossil fuels are getting limited, and there is a global energy demand keeps on increasing day-to-day activities, and CO2 release from fossil fuels cause climatic change. Another important factor is because there is a lot of climate change is a major factor along with that, and these generation biofuels are not only able to generate the biofuels, but also it is involved in environmental cleanup. And basically the lipid petroleum transport fuels are hard to replace by any other renewable sources like wind, solar, and hydro. Wind, solar, hydro have their own positive points, but still liquid petroleum can be only replaced by biofuels. Finally, biofuels are carbon neutral or a negative. That means the net carbon input and output of carbon should be zero. So they are called carbon neutral fuels, and that's why you are not trying to put back some energy carbon into the atmosphere, which causes climatic change. So in that way, these are other carbon neutral or carbon negative processes. So the reason why that's why the reason we need biofuels essentially as a replacement or alternative for the fossil fuels, which has a huge demand. So this is a very preliminary slide for the graduate and undergraduate students, because you have to understand what kind of algae we're talking about. And as I said, I'll be talking more on third generation biofuels, and that means that there are different kinds of algae, and broadly they are classified into three different types: cyanobacteria, micro and macroalgae. So the picture here we are trying to see is cyanobacterial. Cyanobacterial may be filamentous, there can be single cells, and I think most of you guys know cyanobacteria, very popular spirulina, which is a single cell protein, which is a high content protein, which is very well-known commercial product which is available. So cyanobacteria, in some sense, they are the primitive evolutionary of bacteria, and cyanobacteria as an endosymbiotic theory where the cyanobacteria, microalgae, and macroalgae has been evolved. And these are especially the photosynthetic. So they are blind. Also, these algae can be categorized based on the color spectra. That is, blue-green algae is cyanobacteria. They are green algae, which is called microalgae, and brown and red algae, which are primarily macroalgae. So the second type of algae, which is very predominant, and most of the research is being done on diatoms and microalgae and green algae chlorophytes in members, where you can see there are different kinds of microalgae which are present, and there are multiple types of microalgae which are able to produce biofuels. So these are some of the very microscopic pictures of freshwater and unicellular marine microalgae, and also some diatoms which are very important, and we'll be talking more about these microalgae. So our group is interested or trying to solve the biology of these microalgae especially. So we work on other systems also, but basically our more strength concentration, we try to concentrate more on microalgal omics especially. The final one is macroalgae. Macroalgae is very popular. It's known for food and feed like you use in food, like seaweeds. Very popularly known as seaweeds, and seaweeds are also used as a very many important products are available from these seaweeds, especially from brown and green algae. So they are huge collection of huge diversion of species available, diversity of species available of in algae, and that depends on you what is your end product, what you are to look for. So if you and also they are mainly categorized as two types of algae. Either they are known as carbohydrate rich algae or lipid rich algae. So you want to produce an algae for biofuels, then you should select an algae which is having lipid as a resource, rich resource. Whereas you want sugars or carbohydrates for fermentation. For the fermentation, you have to choose carbohydrate rich strains. So based on that, you can do a preliminary assay where you can quantify and you can understand what kind of algae you want for the future, like for your research of your interest. So that's basically broadly dividing the classification of microalgae. As I said, there are different generations of biofuels which are we're looking, and we're talking about third generation biofuels, mainly comprises of algae. So algae, why algae? Because algae, they are having huge advantages, such as amount of oil productivity per acre than any crop plants. And this is a very popular known thing where the amount of oil by algae which is produced per acre is always more than any of the land plants. Thus, it's also seen that you can use microalgae on non-arable land. A variety of water resources can be used. What are those? Like you can use in marine waters, estuarine waters, you can use in waste waters, you can use in fresh water, or you can use any kinds of variety of water to grow that. But you can use these are mainly can be used in non-arable land, thus solving the issue. This is food and fuel. Also, microalgae are important in mitigation of greenhouse gas emissions. As I said, that it not only produces biofuels for you, it also tries to it tries to solve the issue of environmental hazards. So they are very important in clean energy projects also. So that's why they are very important in terms of, so you can convert the hazardous or toxic substances into very useful by the medium of algae. As I said in the starting, it has a great potential not only in producing biofuels, but also valuable co-products. So in that way, you can produce many number of products like lutein, astaxanthin, the omega-3 fatty acids, which have a higher commercial value in nutraceutical and pharmaceutical industries. So thus you can produce not only biofuels from this algae, but we can also produce high value products from these. That's why algae has significant importance in the area of biofuel research. But still, we can say that we know much about the algal physiology and genetic engineering, but still there is a road ahead to really establish to a commercial scale where we can take it to the leads of an industrial production. So in order to just to understand how a microalgal biorefinery can be defined upon, this is a pictorial representation. It's a very popular publication in American Scientist, which has come in 2011, and it's very interesting for a graduate and undergraduate students or anyone to read it, where they are new to the algal research. So where you can see that it requires primarily three components: water, CO2, and nutrients. So you have an algal pond here on to your left side, where it is shown as algal growth, where you can grow the algae with minimal nutrients, water, and carbon dioxide, which can flux into that, which can be utilized and converted. So water is evaporated and once the algae is grown, you can separate the algae using different techniques, either through flocculation or settlement, or you can use any of the chemical coagulation methods and or dissolved air flocculation, where some microalgae has the advantage of floating onto the surface, where you can separate from the water, simply separate from the water. The third one is centrifugation, which is an expensive process, but there are methods and where you can use solar energy as a electricity producer for centrifugation, where the net energy input will be minimal. So this is a big, huge area of challenge, how people can look upon, where how to separate the algae from the water, because algae is grown in water, and the huge challenge is how to separate or what you call as the extraction, what you call as the separation of algae from the water. So this is the main process where it requires a lot of inputs and research, and flocculation is also one popular technique which can also have, and people are working on different flocculating agents which are cheaper, and it can, and some of the algae also have symbiotic bacteria which helps to flocculate. So people are working on algal bacterial, algal fungal interactions for flocculation, because these bacteria or fungi which are produced can produce some chemicals externally which helps the algae in flocculating the things. That means they can get separated from the liquids. So that's this is an area of concern where there are different, basically we see the whole biorefinery approach as the 1, 2, and 3 stages, where you have first the culturing conditions, then separation, then extraction, and finally the products what you get in. So thus you have different stages of that research people are working upon to improve upon the conditions, but still we are way far away to make biofuels entry-able through algae, but still a road ahead is possible. The other one, the third phase is extraction, because you have an algae separated from the water, and now it's very important to how to extract the lipids or high-value products what you're looking for. So there is a different process where you try to extract using different polar and non-polar solvents, and that phase separation, and the lipids can be coming into the polar solvents, and from there you can extract the oil, and that can be used. That oil can be used as a raw oil, which can be used as a biodiesel, as jet fuel, naphtha, by upgrading through the hydrotreater. So biorefineries already have these processes. So once you get a crude oil or a raw oil, that can be upgraded using a hydrotreater available in biofuels, and you can produce different products at the end, like biodiesel, jet fuels, and jet fuels or nothing but a small carbon chain molecules. And during the extraction process, the important thing is whether you can able to solvent recycling or use the green solvents, economical solvents such that that can be used, and the sludge or a spent algae or water which is being coming can be recycled with the minimal nutrients back, and that can be put back into the algal growth media for the growing of algae back, and with the addition of that, and the biogas which is generated from the overall process can be used to run the turbine that can be generating the power for supplying for the algal ponds or algal open or closed reactors. And the sludge can be used as a biofertilizer or anything. So anaerobic digestion can be. Thus, you have a multiple products with a single system which you are trying to look for that. So thus, and aggressive recycling is essential to make the algal biofuel sustainable. And these are the steps which were involved in looking for the biofuels, and that is how you can really look forward to make algal biofuels sustainable. So because when I discussed different steps like culturing, extraction, or separation, and finally the end product, there are still challenges which has to be looked, where I said like large scale production. So it's a huge challenge. What kind of nutrients you have to supply? How do you recycle that? So how do you put the gas transfer and exchange? So if you are trying to put the carbon dioxide, how you are going to get it from there? And you have industrial dioxide or a flue gas which can be used for the algae to grow. And the very important is photosynthetic active radiation delivery, because these are green photosynthetic organisms and requires photosynthetic efficiency. Culture integrity, so you have to work on these things. Environmental control, that is very important. Land and water availability, and many more. So these are the challenges which are there in algal biorefinery, and that has been sorted by many people and in due course of developing those things. Regarding the harvesting, as I said, we can undergo the flocculation, filtration, followed by centrifugation or a drying process. Drying is natural drying, just keep in the sunlight, and that will not cost you anything, but only the time. So there are different processes where you can try to harvest the cells. Presently, the cost of a per barrel is very high, and also the overall productivity due to all these challenges is making the biofuels not feasible. So that's the reason why the high cost of biofuels is being reflected. So if you can match the processes where you can produce biofuels at the rate of the fossil fuels, then the algal biofuels or any kind of biofuels are really compatible or competitive enough to find an alternative solution, which is the future of what the improvements we are trying to look forward. So this is a dream come true, where this is a small video or an advertisement from ExxonMobil. This is a very catchy phenomena where why algal can be looked as an alternative for biofuels. And I'll just play, this is just for 1 minute, and I'll try to play this video first. So this is a wonderful example of how algal biofuels can be used in the context of generating the next generation biofuels. And it's a very interesting small video by ExxonMobil, and it tells you everything about what we are looking or anticipating with. So basically, our research group, we call it as B3 Algae, because B3 stands for biomass, biofuels, and byproducts, which are the three important end products what we are going to look forward. As I said, this is a pictorial representation of algae, how it can take three components as inputs and can process it in an algal system, and the outputs will be biomass, biofuels, and byproducts. So basically, the sunlight, any kinds of gas like industrial waste gas or atmospheric CO2 is used by an algae, is taken up by the algae in the presence of in water like wastewater, fresh water, or seawater, and that will be processed in the algae to produce biomass, biofuels, and high value products, along with other hydrocarbons which can be used. So the biomass can lead to animal feed, biofuels, heat and electricity, and biogas generation. And the biofuels or the biodiesel precursors which we are looking for, and the major concern now is to look for what are the basic things which we can lead to biofuel production and high value products, like as I said, like antioxidants, tocopherols, vitamins, antioxidants, omega-3 fatty acids, astaxanthin, lutein, beta carotene, different things can be produced through these things, and which has a significant importance in pharmaceutical, nutraceutical, and other industries. And it can be used in production of electricity, which is used directly into the photovoltaics, and people are working on this system. Recently, we have come up with a paper in bioresource technology with a collaboration with other university, where we try to use algae as a substrate and purify the wastewater, and that also generates, and along with the biofuel, it is able to generate the electricity. That does, as I said, like algae has a potential to not only produce biofuel but also other high value entities. Also hydrocarbons, algae, some of the groups are working on generation of plastic and the other petrochemical substituents, which are very important sources in some of the algal systems. So thus we can produce multiple products. Thus we can produce multiple products for algae, which are very much useful in that. So mainly this is a very translational process where you use three major inputs like sunlight along with minimum nutrients, carbon dioxide, and water, and the algae processes that into different systems. Not only algae can just produce these products, they are also involved in treatment of wastewater and industrial waters, or any kinds of waters that can be converted into potable. This is a part of mission innovation programs in India around the world, where we are trying to look on different aspects of how we can purify water using algae. So this is a very, very relevant area where people can look upon how algae is able to do that process. It's doing already in nature, but we are trying to understand more in that. So as I said, there are important paradigms to understand the algal biotechnology, and here the one main thing what we are trying to understand the technology is through different approaches. So this is an example where you want to produce the biomass and lipids together. So when an algae is, so there are two different strategies where you can try to improve on those conditions and to produce biomass or lipids. So the most popular techniques which are used in nitrogen is deprivation. That means algae is present and you try to remove the nutrients from the algae. They are producing lipids or neutral lipids as a process of getting stressed up. So in that process, the biomass is highly compromised. So we need to have the biomass along with the lipids. So we have to know how to adjust those factors. So another important factor which people work on is genetic engineering. So you can put a known or homologous or heterologous gene inside one system and try to overexpress that gene of interest, where you can increase the lipids or other high value product. So all these techniques have ups and downs, as every process, if they are trying to divert the carbon towards something, the flux tries to compromise on other things. So it's important to have both together without compromising growth. That is the factor we are looking for. So the other side of the research, what we are looking is whether we can increase the particular biofuel or without doing anything, biofuel or biomass of, without compromising the biomass. And the major thing, the strategies what we are doing is media engineering. So media engineering is an approach where you can just try to modulate the environmental factors, because microalgae is already producing these substances but at a lower level in the natural environments. So in order to make it industrially efficient strain, what are the methods you can apply that you can slowly increase the media, decrease the media, plug and play with the different nutrition parameters, so that you can enhance the biomass and lipids. You can enhance the lipids and high value products without compromising the biomass. Other recent technique which has come is chemical modulators. These are nothing but it's a well-known approach in plant systems where you use growth promoters for enhancing the growth in the plants like auxins, gibberellins, and all those things. Whereas the chemical modulators, this is a phenomena, is a small molecules, organic molecules, which can be used in the media where you can enhance either lipids without compromising the biomass. So this area of research is been in under development, and people are working on different molecules. But the problem is whether using the chemical modulators you are increasing the cost or not. But there are some certain researchers where research perspectives where they have shown that we can enhance the lipid without compromising without compromising the biomass. Thus, we can. So they still there are processes how we can really subject it to that, and they are kind of 1 stage and 2 stage. And till date, I think the best method is to first enhance the biomass at initial stage, and the second one is to subject it to nitrogen deliberation so that you can produce high number of lipids, or based on what you are interested in, you can produce that. So a 2-stage approach in microalgae is more promising, and people are working on that. And in our lab also, we work on different aspects of 2-stage approach using media engineering, CO2 supplementation, chemical modulators to enhance the biomass, lipids, and the byproducts. So in order to understand this, how we can really understand what is happening within the cell is more important. That is the basic research, which is very, very important for any researcher to understand how to modulate your system to produce your product of your interest. But in order to do that process, you have different approaches. One is omics research. What we are doing, it is at a different level of stretch at multi-omics platform. By understanding different aspects of omics, the interior physiology or metabolic networks of the cells can be understood, where you can apply through genetic engineering to improve that the processes, along with pathway engineering. The most popular and transcriptional engineering. The traditional way is you will knock out a particular gene or 2 genes, or you will overexpress a gene in order to produce that. Now the new modern way is transcriptional engineering, where you can find out the transcription factors which are important, and you can knock down the non-essential transcription factors so that you can divert the carbon flux towards the any product of your interest. For example, there are some transcription factors like BZIP and H1H. These are the factors which are involved in production of lipids. So if you want to, if you knock it down, then the carbon flux can go towards the carotenoids. So it depends on the product of interest. You can do the pathway engineering or transcriptional engineering so that you can produce a product of interest. That's how all these omics data or engineering data can be put in systems and synthetic biology. And before generating before understanding the systems, we can try to understand more in detail how you can substitute all the data which is coming from the omics into the model, and that will give you an answer whether your model will work at a other level or not. So different conditions can be where you can input all the data into the model and see through metabolic models and genome scale models whether the particular thing can work at an industrial level. So all together will give you the rationale towards reconstruction of photosynthetic cell factories, which are able to produce the important product of interest. So this is how the paradigms in algal biotechnology can be answered at a molecular level. So to understand different aspects of that, so that's how we can go ahead of that. So today I'm going to talk on a small case study what we are doing in a lab, where we are trying to understand the cellular dynamics of microalgal cell factories under nitrogen stress condition. I will explain you what is nitrogen stress. As I told, when there is this deprivation in the media, it tries to accumulate towards lipids, which are called neutral lipids, which are the biofuel precursors. So how we are trying to understand that phenomena using multi-omics platform to identify relevant enzyme encoding genes and evaluate these mechanisms in these oleaginous microalgae that will contribute for the engineering of regulatory networks for increased oil accumulation. So in this case study, I'm going to discuss about how we are trying to understand the regulatory networks of algal strains, and in future, how you can fine-tune these networks so that you can produce the biofuel or high-value product of your interest. So this is how we try to understand the stress biology in microalgae. Basically, on my left, you can see that nitrogen deplete condition, where basically when nitrogen deplete is a condition where you try to remove the nitrogen from the media, and which is a major component of all the important essential biomolecules of a cell. Basically, what happens is all the primary metabolic activities like photosynthesis, beta oxidation, all these other cycles are reduced or are downregulated, whereas the lipid biosynthesis goes up. That means the accumulation of tag bodies, which we call it as oil or lipid bodies within the cell, happens. And this is a very popular phenomena within the microalgal cell factories, which is otherwise known as oleaginous microalgae. The microscopic picture in the middle shows that the cells are green in color, and there is a yellow background within which are called oil bodies. So when you strain with it with Nile red, which is a fluorescent dye, you can see the orange fluorescence. That is nothing but, you can see after the particular 6th or 10th day, you can see that the cell consists of only the neutral lipids or oil. But there is a huge compromise on the biomass in this study. So we are trying to understand what happens when it is a nitrogen deplete within the cells. So that is how we try to understand what happens at the transcript level, protein level, and metabolite level in order to understand the biology of the organism in that. And on to my right, there is a replete condition. That means that once you have the cells, initially the cells are in replete, means nutritious control condition. We grow them and deplete those cells and try to put back the cells because this is very important. The basic phenomena of understanding this is because once you milk away the oil from the microalgal cells, when you want to make the cells active again, because they are under stress, so you supplement all the media with your nutrients of interest, then the algae starts growing within 12 hours to 24 hours into normal cell. That means it gets relieved of the stress, lipid biosynthesis goes down, that is the accumulation of neutral lipids goes down, photosynthesis, beta oxidation, other central metabolic pathways comes up. This is a very important phenomenon, and it's a very quick phenomena which happens within the microalgal cell. And if you can develop a system where you can produce a lipid of your interest, milk out the lipids or high value products from the system, and supply for a couple of days with the replete condition, then the cells are again replenished with your fresh media, and they get activated into normal cells, where again you can, so that's the process will become continuation process, and thus the feasibility or sustainability of algal fuels are more promising. Thus you will have a complete cycle process of how to generate the oil and to extract the oil and all. At this stage, we know we can grow the algal in 2 stages, and we'll extract the oil, and the algae is left alone behind as a biochar or a biofertilizer or a protein-rich biofertilizer, which can be used on a feed and food industry. So this is what the phenomena we have now, but there is an opportunity where research can be done where you can milk out the oils which is present in that, then you take back the algal cells back into the fresh media and replete them back into the normal cells. That thus you can use the same system, recycle not only the nutrients, recycle not only the extraction solvents, you can recycle the microalgae, which will be more promising and all. So in order to understand, you have to understand the biology of the system. So this is one of the phenomena we look in different dimensions of that. So in order to understand that, we look for the stress physiology of the microalgae, and this is an example recently published in Biotechnology for Biofuels, and this is where the topmost left figure shows the growth curve of the microalgae. It's an indigenous pollinaceous microalgae called Parachlorella kessleri, which can accumulate around 50% of dry cell weight in nitrogen deprivation. The oil can be accumulated or lipid can be accumulated. This is a typical example of pollinaceous microalgae which can be produced. But the process is when you remove the nitrogen, the cell growth is compromised. The red line is when you remove the nitrogen, the growth is highly compromised, and the blue or green light lines are the ones where you see that the biomass is not compromised. The green one is the control and the blue one is the phosphorus deplete. So there is a unique phenomena where in these cells, particularly these marine microalgae, if you try to remove the lipids, but the cells does not, the cell growth is not compromised, but it can even produce high amounts of lipids. So this is a promising phenomena where the cells are able to accumulate high amounts of lipids along with no compromise in the growth. So certainly this microalgae can be fine-tuned for the production of biofuels in near future. So the right one on the top shows the TLC of how the lipids gets increased. The TAG, what it is named here, is the triacyl glycerol or neutral lipids, which are present as a form of lipid bodies, and this is mainly consists of triacyl glycerol, and these triacyl glycerols are increasing. So 0 to 10 days, if you see that in nitrogen depleted conditions, on second day the oil gets accumulated. By 10th day, the cell is completely full of oil. The same thing with phosphorus depletion, where you can on 10th day the large amounts of oil is being accumulated in form of neutral lipids or tags. In sulfur depletion, there is a compromise in growth, and also oil does not increase because it's not a sulfur-rich isolated microalgae. So that depends on the environment where you are taking that algae from. That's why, but the sulfur also plays a major role in that. But there are, in this particular microalgae, we didn't see much changes, but there is a huge compromise in the growth in terms of sulfur depletion. So on the bottom left side, we can see the FAME content. So you can see the FAME content on the control is very low at 50 microgram per mg per dry cell weight, where in lipids it almost increases to 300 microgram per gram dry cell weight. So roughly it is almost increasing 50 to 300. That means it is almost increasing to 5 to 6 fold increase in that. So likewise in phosphorus deplete condition also you can see that there is a huge increase in the FAME content. But the productivity goes down due to the number of cells. There is no biomass. So the overall productivity goes down because the number of cells is more, but in this the productivity goes up. The number of cells is lower. Whereas the sulfur shows even there is increase in FAME content, but there is a high compromise in the biomass. So, and also it is very important to understand the different profiles of these fatty acids, whether it consists of saturated fatty acids, monounsaturated fatty acids, or first, which is polyunsaturated fatty acids. And we see that the nitrogen has more of saturation and monounsaturated fatty acids, which are important for the biodiesel precursors. Whereas in phosphorus condition, you have more of polyunsaturated fatty acids, and you know that because of the growth of the cell, the more of membrane lipids are being produced. So in that way, you should have, if you want a better biofuel precursor molecule, you need a saturation to monounsaturation ratio in a higher level than PUFAs. But you need a little bit of PUFA concentration so that cannot. So in overall, there are different processes where you are trying to understand, and when you understand the basic biochemical constituents of a cell, you try to understand what is the ratio of carbohydrate, lipids, proteins, carbohydrates, and carotenoids in overall when it's subject to any particular condition. And you see also the individual components like polar lipids, saturated lipids, monounsaturated, and polyunsaturated lipids in that course of time. So in order to understand, as I said, we would like to understand the whole system using an approach called omics research. And this omics research not only gives you the physiology of a cell, but also it can gives you the phenomena, what will happen within the cell after a particular when you try to modify a system or try to subject to physiological condition. This is a very popular technique nowadays, and NASA is using omics technique to understand the astronauts' physiology because they provide these astronauts with different food, nutrients, and all those things, and they try to see what is happening to their protein metabolite and other contents of at a physiology level, so that when these astronauts are going into the space, they can there will be no change, and what the changes they are going to expect from that. So a journey to see more than ever before can be answered through omics research, and this is not only applied for a microalgal. This can be applied for, it is a technique that can be applied in many research perspectives. So omics research is unique in its own terminology. So this is especially a vocabulary for graduate undergraduate students who does not understand much on omics. Basically, omics aims to the collective characterization and quantification of pools of biological molecules that translates into a structure, function, and dynamics of an organism or organisms. So basically, it will give you a physiology or an introspective of interest in what is happening within the cell when you are subjected to particular physiological changes. It may be environment, it may be any kind of stress, it may be anything, starting from a single cell, bacterial cell, to a human, highly evolved human. Omics research is very important to understand the health. And the future of science is through omics, because if you are able to understand the physiology of a human being, the personalized medicine aspect of near medicine in future will be based on omics research. So you can now understand what is happening within at a gene level. You can be given a personalized medicine based on that. So this thing, what kind of drug you should be given, or all these things in near future will be depending on that aspect of that. So basically, as I said, like omics is a study of ecosystem, including bacteria, virus, fungi that can keep our body and give our this thing. And genomics is a study of DNA within a single cell or an organism. And transcriptomics deals with the RNA, which is important, where the genome genes get transcribed, and transcription translational into proteins, where the study of proteins is called proteomics, and that change in the protein level is causing the disease in a particular organism or a condition within an organism which can be recognized. So epigenomics is a study is getting popular, and it's an integrated study where some like to understand, or a part of can be expressed and see how what are the changes between them. And metabolomics is the one which is happening within the cell organ, and the organism helps to maintain the life, and thus monitor the process of metabolic within the cell. Thus, omics is having multiple roles to understand, and each level has to be understood in order to have a consolidative approach of understanding the physiology of the cell or a system. So we call it as integrated omics or iomics. So basically, iomics is nothing but we know that if you try to use one omics like transcriptomics, genomics, or a proteomics, or any single omics, you try to only understand only at a particular level, but you cannot understand what is happening at the next level. It is very important to understand from the genomics to transcriptomics, from transcriptomics, proteomics, and metabolome, what is happening within the cell, so that you can establish the final aspect outcome of the cell. So in order to understand, single omics will give you within the single layer of cells or cell factories, which what is happening between them, but it cannot correlate, because you know that whatever protein is getting translated can be whatever RNA is getting transcribed is not translated in protein, and that protein is not converted into metabolite. So in order to understand, we have to focus on one or two molecules or more than one this thing, which we call it as a multi-omic approach, where the genome or a proteome can cross talk with the metabolome, or a metabolome can talk with transcriptome, in order to understand the pattern and the outcome of that. So an integrated omics or iomics approach, integrating all the advancements, is to understand the molecular mechanisms of the cell. So in our lab, we do different kinds of omics approach, just like forward genetics and reverse genetics. We call it as forward omics and reverse omics approach. When we start with genomics, so we have the DNA, we transcribe that into mRNA, and then proteins then go to the metabolomics of the cells. Then that is called forward omics, where you have the thing, but the latest the approach is reverse omics, where you don't have any information on the genomes or transcripts of what is happening within the cell. Like many, as I said, there are many species or strains of microalgae which are available, and we don't know many of those cells of microalgae, many of those behind that, what is happening behind them. So in order to know that, we can go for a reverse approach of reverse omics, where you take the cells subject to different conditions, compare your metabolomic profile, and that can be correlated, and that can be mapped on the ICGEB mapper, where you look on different metabolites and correlate with the proteins, then proteins to transcripts. Thus, you can come into a consolidated picture what is happening, and you can also modify a particular process using these approaches. Thus, integrative omics approach will give a cumulative picture about what has to be understood on the molecular mechanisms within the cells. So in order to understand these experiments, so there should be mainly three important factors, very important for any researcher to start with. These are what we call an experimental design for omics research. So what are these? The use of suitable biological samples. The choice of sample type to investigate is dependent on the objectives of experiment and the availability of the sample. You have to have a reproducibility, so you should have a sample type so that you can investigate. You cannot do just for once or once. You should have a suitable sample in order to understand. That means you should have a time course or a kind of a condition in order to compare what is happening within the thing. So that is very important to set up your experiment based on that, and you should have a technical and biological replicate. So it is very important that, for the undergraduate students, I want to differentiate what is a biological variation and a technical variation. Technical variation means if you have one sample, you run the sample for 3 times, that is called, from a single sample, that is called technical variation or technical sample. So if you want to see a variation, you should take a sample 3 times and run on an analytical instrument to get a thing. And biological variation is the one that the same condition you have to run on the cells or a system for 3 times, and minimum 3 times, and you have to get those things. So that biological variation and technical variation is very important for omics study, which has to be considered. So these 3 important. You have to design your experiments in a time course way such that you can understand the system better, what is happening at a particular condition. The second one is either you should have it is mandatory you should have a technical and a biological variation of the studies, such that the changes which are not found at one instant can be found out. So there should be a collective effect there, which should generate a metadata, which can give a strong comparison between the groups, and it will produce. So these are mandatory when you are designing your omics experiment, and it is very important to understand this thing. So in my case study, I'm going to give you a glance about the reverse omics approach. Excuse me. Reverse omics approach, where we try to start with, or as I said previously, so we start with metabolomics and we end with the genomics. So the same thing. So what is metabolomics? So this is especially for students. There are two types of metabolomics. You can use either targeted based metabolomics or untargeted based metabolomics. Basically, when you don't know anything, you go for an untargeted approach where you get the global metabolic profile of a sample. That means you have the cells, you have the tissue, you run the whole metabolite extract from LCMS or a GCMS, which is liquid chromatography based mass spectroscopy, or gas chromatography based mass spectroscopy, such that those data can be analyzed and validation of MS using standards, which can build up a global metabolic profile of the biological samples. So this is an untargeted approach, whereas in targeted approach, you know what are the metabolites which is being produced after once you do the global metabolite profile. And in targeted approach, you select only a particular metabolite and you try to use the standards, external standard. First, you run the standard as a metabolite and you know where the standard is coming based on that standard quantification and quantification. You run the samples and you see what is the change which is happening between the things. These are the most popular tools or techniques in metabolomics. Either it is targeted approach or untargeted approach, or it is a reference-based approach or a de novo approach, what we will be doing in the omics thing. So for metabolomics or proteomics, we call it as a target-based or untargeted based. Now I'm talking about untargeted metabolomics because this is a new strain where we don't know anything about the genome. So what we'll do is first we try to put all the, just extract the sample and take all the metabolites of interest through standards and data analysis and put it on the CAG mapper to do that. How do we do that? So it's a simple thing. As I said, the experimental design should be the same one what I have discussed earlier, and you can do the sample preparation. There are different processes like derivatization when you do the GC based approach or a gas chromatography based mass spectroscopy. You will do the sample preparation by derivatization using different silylation, alkylation processes, and you try to silylate or alkylate a particular metabolite, and that can be quantified through GCMS. So if you're doing liquid chromatography, this is LC based MS. You just directly extract the sample and take the sample and inject into the machine, and that can be separated based on the mass. So NMR is the one more technique where you can do the metabolite, but the drawback of NMR is you cannot do untargeted, but you should know what you are doing it, and you require high quantities of sample when you're processing. All three together, you can you can do the pre-processing of data, identify the metabolite through the library, and that metabolite can be put on the KEGG mapper, and this KEGG mapper will give the pathways which are involved, and that's how you can hypothesize and validate your hypothesis, what kind of metabolites are regulating and down-regulating based on the process. This is the overall process. How do you do with the simple metabolomic process? And that is how you can take ahead with that. So this is how we have done with the sample. So as I said, like when we were subjected to nitrogen starvation, we have run the metabolome profile of that, and there are different metabolites which are involved. We have compared with the metabolites which are upregulated and downregulated. And these are the different data analysis where we have seen that there are number of metabolites which are upregulated, 5-fold change or 10-fold change. Based on that, we come to the hypothesis and we map it onto the KEGG mapper, and that can be done. So on mapping into the KEGG mapper, we have come through that. There is a very important, so you can see that nitrogen is labeled in N wherever there is an increase. Phosphorus is in wherever there is blue, there is a decrease. And nitrogen is in red, phosphorus is blue, and sulfur is starvation is in yellow. And the arrow indicates the upregulation and downregulation of particular metabolite. So we have seen that the overall flux towards the carbon flux towards the fatty acid or attack biosynthesis is through the acyl-CoA pathway, where the red line shows the upregulated pathways and the blue line shows the downregulated pathways. This shows that there are a lot of pathways which gets downregulated in nitrogen starvation condition, and all the carbon flux is diverted towards the lipid biosynthesis. This has been recently published in Biotechnology for Biofuels. So I'm giving the niche of every omic technique what we do at the lab, and so you can understand. But to in detail, if you want to have discussion, there are a lot of things, and you can contact me. But each technique requires a lot of thorough understanding and a lot of standardization of the sample. So this is regarding the proteomics. Proteomics also the untargeted and targeted approach. This is again, you have the cells or a tissue. You extract the protein using different methods. We use trizol method for microalgae, where you can extract the method, and we have developed a method like from a single sample of microalgal cells, you can extract the DNA, mRNA, and protein from a single sample. So that is a very efficient method, and you can use it in your lab also. The other method, once you extract the samples, you have to separate through any of the chromatography process. The popular is LC-MS based method or BitRAP based mass spectroscopy, and you run the data and you look for the proteins. So when you get the peptides, digest with trypsin, and when you run on the LC-MS based approach, and you analyze the data through any of these softwares available or a library. So you can map it to using your reference-based genome, where you can compare what are the peptides you are getting. This is the basic principle, just like metabolomics, proteomics. These are all very common. So extraction, separation, just like in metabolomics, and finally analysis through enzyme. So basically, your LC-MS mission will separate and give you a mass can be compared what kind of peptide you are getting or protein you are getting in the sample. So this is another popular example where you can see that you have a sample which is run on SDS page. That SDS page is excited, the proteins are taken, and trypsin digestion is done, and you have a mixture of peptides that is run through LC-MS ESI approach, where you have different mass by charge ratios that can be seen, and this can be extrapolated what kind of amino acids you are getting. So the mass by charge ratio gives that. So this is the overall process how the proteomics can be done, and whether it is a targeted or untargeted, you can do that. And proteomics is the one way where you can understand all the phenomena changes within the cell, and because that is the one which is having more and more, and it's more complex than metabolomics approach. So this is our sample where we try to run the peak accelerator subject to nitrogen separation, and you can see there are almost 1763 proteins, which were significantly 166 proteins were upregulated and downregulated in course of when it is subject to nitrogen solution. So we try to overlay the metabolome data with the proteome and the transcriptome and the genomes together. Thus, it is called a multi-omics approach, and it is a first of a kind of where we are using multiple omics approach to get a conclusive picture on that. The third important technique is a transcriptome technique, where you have different transcriptome analysis of mRNA or RNA-seq technique, where you use either Illumina-based or ion torrent-based or solid-based. These are the chemistries behind that. I will not be discussing on each of the chemistry because you can get it anywhere. Because based on Illumina, you can get a short read. You have other techniques where you can get a pack bio or anything. You can get the long read. So it is mainly the genomic approach. So if because RNA is a short read, it is more of ion torrent, solid, or ion Illumina. So the most popularly used technique is Illumina technique, where you have beads tagged with the fluorescent tag. So once a particular nucleotide is found out, that will be signaled, and that's how the reads are done. So you get a small reads of 75 into 2, 150 into 2, or 300 into 2, different on that. So the popular technique used for transcriptome is Illumina, as ion torrent and solid are little bit expensive or in a stage of development. There are different companies which can give you transcript mRNA or RNA-seq data, but the most popular is Illumina, and the other companies are also coming up with that. So what we have done is, you can see this is the pictorial representation. We have different transcripts on us, and the red and blue shows that we have integrated the genome and transcriptome. The blue is the one and the overall pink pathway is the one which are all the metabolic pathways. So because we were initially interested to look in what is happening for the lipid biosynthetic pathway, why these neutral lipids are formed. We tried to pick the important genes of interest and tried to correlate with using qPCR, and we have seen that there are many genes which are downregulated and upregulated, and that data is correlating with our transcriptome data. Once you get the transcript data, you have to correlate number of targets by qPCR, and you see whether whatever fold change you are getting in the transcriptome data, because transcriptome will give you huge number of transcripts, overall transcripts, number of transcripts in that. But you have to look forward, like what are the changes which is happening with the qPCR. So it will be a selective approach where you can do that. So our transcriptome data shows that so the number of transcripts which are upregulated is shown in red, and which are downregulated in blue. So and there is no change with the black ones. So you can see that there is a red line which shows that an increase in fatty acid biosynthesis that is leading to oil production. So that's how you can map each process which is happening with a particular condition, and you can find out the solutions, how a particular high value product or bioal is being generated, and how you can mimic the conditions. So you can also see that the whole TCA cycle is not active. There is a partial TCA cycle, what we call it as half TCA, where the malate is being recycled into and it is generated into pyruvate, and the pyruvate is the precursor for production of this thing. So we can see that overall process, and this is very similar in most of the microalgae, and you can also look forward, because omics will give you a complete global scenario of what is happening at different levels, and you can integrate those data. So this is the overall complete integration of genomics, because genomics of this particular strain is done at ICGEB using a novel dual sequencing or a hybrid genome sequencing technique, where we could map it onto 396 scaffolds, where it consists of 8,000 functional genes. So this is information which is very much important for us to understand and as a benchmark for the other studies. So integrating all these things, you can see a complex picture like this on the KEGG mapper, which shows you how the pathways are connected or interconnected, what kind of pathways are getting upregulated, downregulated. All these scenarios will give you the individual picture of what is happening within the metabolic pathways in a particular condition, and how to fine tune these cell factories in order to achieve whatever you are looking for. So in final testing, what we are trying to understand the biological systems through integrating the omics to engineer cells is through iomics platform, where, as I said, like it is a powerful technique which can be regulated and connected with a number of feed forward and feed backward mechanisms that can be positively or negatively ultimately affect the system's output. So thus, in understanding at iomics will lead the identification of relevant enzymes and reconstruction of metabolic pathways involved in biosynthesis and degradation of potential production. That's aiming for vision for tomorrow. So if you're understanding the multiple data sets, that data sets can be put into a model which can stimulate how the system can work it out. And also you can engineer the particular pathways so that you can also understand that. So these all together will give you a concerted effect of understanding the microalgal cell factory at a laboratory level, which can be mimicked what will happen when you take that strain to the large scale production. So all this understanding is very important, because each microalgae, each system behaves in the own way based on the nutritional conditions, based on the support system you are giving. So based on that, it is not the common that every cell will behave or every microalgae will. So it's very important to understand the system into the core of that, so that you can modify those systems on how you can generate these different byproducts. So finally, this is the fourth generation biofuel, where mainly, as I said, like fourth generation biofuel consists of fixing up the CO2 by microalgae, where it can be converted into solar system. that can be separated on a non-agriculture land. And this is a kind of a diagrammatic representation by Joel technology, where they have shown that how you can use the microalgae to capture the CO2 or using sunlight. That can be used to produce by separation, and that can be used for fuels and other chemicals in the due process. Thus, engineering strengths, because engineering of the third generation biofuels is important so that you can take the advancements into the fourth generation biofuels. This is a nutshell where you can use the very many important processes like solar in terms of hydro and all these things integrated together to develop a fourth generation biofuels direct to product to process for any kind of a feedstock you require. So that's there's a dream come true in next few generations, and the people who are now graduate, undergraduate students or postdoc students may be taking up in this field of research where it can be really essential for the whole global community. Finally, the case study, because this is very important, whatever we are talking about algal biofuels, is it very feasible or sustainable? Yes, the answer is yes. This is a very good roadmap study which has come up with a team in 2016 NAVB report synopsis, where you can, if you are interested, you can just go through this, because what happened is the consortium of pool of scientists, many number of scientists around the different universities who are working in this, and they have been working day and night, like almost for a decade, to understand this roadmap and build up this roadmap. In this roadmap, they tried to screen different microalgae, and they selected one microalgae for further. They developed the cultivation process. After the cultivation process, in a high-scale model, they try to harvest through flocculation, which after flocculation, they try to produce the, and overall, the overall net process shown that you can produce the overall biofuel at $7.5 per gallon, and that can be still reduced if you can find out the solutions for harvesting. And also, they did not add the co-product process. Thus, if you can extract the co-product and go for de-oiling of the algae, then that will be more beneficial in terms of generating a revenue for the biofuel. Thus, it makes it a sustainable solution and also an economical solution in terms of getting down the price of the gallon. Finally, I would like to thank my funding agencies, Department of Science, Department of Biotechnology, Government of India, Department of Science and Technology, where most of the funding is provided by them to do this biofuel research, and Government of India is fortunate enough to encourage us in a lot of these kinds of perspective areas. And I would, without my team, Omics of Algae, a team, so without these guys, it is impossible to work alone. And I would like to thank all my Omics of Algae group members. And most of the work, what I have represented today, is done by Kashif and Asha in my lab when we started. And now we have the molecular tools, and we are happy to even collaborate with fellow faculty around the world in the area of algal omics, and also help to have an amicable solution for that. With that, I would like to again thank the organizers for giving me this opportunity, and the floor is open for questions. Thank you.
Thank you, Dr. Jutur. That was a very captivating presentation. So now I'd like to request our event chair, Dr. Abdullah Muhammad Shual, sir, to share a few words with us about this new platform that he has started and his vision with it. So Dr. Shual, sir.
Good afternoon. It's really a very, very nice and detailed presentation. And obviously, for Bangladeshi audience, it's very new one, as we have not yet started much more work on algal biofuel production or this 4G or 3G biofuel production. So obviously, lots of questions from audience we already received. So first of all, I'd like to thank Almighty Allah that he gives us chances to attend this gathering. Due to this pandemic, lots of sufferings of people here and there, and lots of peoples are affected, economies are affected, education, study, everything is affected. So due all of those, we are here, and then we are doing great. So I'd like to thank Almighty Allah. And then Science Portal BD and Cell Genetics and Plant Biotechnology Lab, those members, especially Saim Nasrutullah and his team, they are working very hard to make this program successful. And then they are really working very hard, like every day, 12, 13 hours of their effort to make this program successful. And then those who are helping Saim Nasrutullah for streaming and publishing and publication and other things. So I'd like to thank all of them. And my cordial thanks to all of the participants from around the world who are here actually for some new adventure. In our last program, we got the similar responses, huge participants from around the world, and then similar things happened this time also. And I am really thankful and also grateful, all of them. So I would not talk much because our honorable Dr. Pavan Jutur from ICGEB, he had made a huge talk this event, from the very basics to advanced. So everything he already described here. So just a few glimpses of the Science Portal BD. Science Portal BD is a new science communication platform. Basically, scientific communication to the people in very simple and easy to understand way to deliver. Right now, our audience is the students, researchers, and other group, because we are almost locked down and we cannot move around. So we cannot reach general people to convey different messages, but we can reach our students and our education facilities or our educated families to send them very lucrative, advanced, and promising messages to them. So we would directly, I am going to the questions. Dr. Jutur, am I audible to you?
Yes, yes.
Lots of questions actually we received so far.
Okay.
Among them, it is not possible to address all of those, but some highlighted question I'd like to raise, and then if you please answer those questions to our audience.
Sure,
so,
sure.
First question, it is possible to produce the biofuel. I mean, this biofuel producing algae can be grown in a eutrophicated pond?
Yeah, basically the idea behind this is you can use the eutrophication ponds, or you can use the marine rich. Say for example, India is having a high marine belt in South India, so you have a huge water body where you can grow microalgae or macroalgae on the shores. So that is also a possible way. And eutrophication, that is where that is very important, because that is how you can purify the water there present in that, and that comes under kind of a wastewater treatment or these kinds of things. So basically you have to work on the composition, because it may lack some nutrients which are not present in the system, but it is so there are two ways how you can look for the eutrophication. So one is you can isolate the algae which is present in those lakes, and you can grow them such that it can be used as a product. That otherwise you can have a consortium and grow in that. So it is both the ways, but it's a very amicable solution, because that is how we can find out the way how to look upon that.
Thank you. And then many questions is similar topic that actually about the feasibility. I mean, this like India, Bangladesh, South East Asian countries, how feasible this technique? I mean, on aspect of economical things,
Okay.
cost benefit analysis and everything. So many
Yes.
question actually from there that how feasible this is.
So basically, as I said, not a solution for the next immediate effect, but we need a solution for next generation. So if we are not trying to act smart and look for the solutions now, tomorrow when the fossil fuels are depleted, we are going to end up in a very, very bad situation where we don't have alternatives and our environment is getting bad and worse and this thing. Basically, keeping all those things aside, but it's very good to have a solution beforehand rather than just sorting out when the problem arises.
Okay, that
And
means just getting, getting the technologies and everything. Whenever we
yes,
need, we can apply. But right
yes,
now,
It's not
it is
feasible.
not much feasible in terms of cost and in
yes.
terms of investment.
Yes, exactly. But there are ways like because there is an alternative way what people are looking for, especially industries or small groups where they started with startups. What they do is they look for high value products, which has a huge
Okay.
market of nutraceutical or a pharmaceutical products, which can be looked initially. Then you can invest the money in a biofuel. So that's how you can look forward to co-extract the co-products initially. That can be a great investment for the further. So we cannot, because as I said, the biorefinery is a huge cost for any government to establish. So can we establish at a ground level the products like high value products which can be used, and that can be taken, that investment somehow can be taken into a bigger companies or a biorefineries that can be developed in due course of time. So that is what the phenomena throughout the world people are trying to look for. But a few like big oil companies they are trying to even jet fuels. So in US they are like Exxon Mobil and other companies have already or in a way to even have biojet fuels. And in India also we have biojet fuels from other this thing, but not from algal source. But they have run the biofuel for airplane. So in that way, we have to look for alternative. And as I said, like it is already hidden in the environment or a nature. So we have to understand and fine tune that so that that will be more benefited for us.
Okay, thank you. And then another question from per kg of biomass, I mean algal biomass,
Mm-hmm.
how much biofuel we can get?
So the basically per kg of biofuel, it is not in terms of that, but usually you can read in terms of tons of microalgae, because you
Okay.
cannot per kg you will not get much of oil, right? So if you talk about some 1 or 2 tons of biomass, then you are able to get around 1 kg of 1 liter of biofuel. That is a good process. So basically, what we try to look is if you can grow an algae at 1 gram per liter per day,
Okay.
and it has a amount around 30% of lipid in that, that will be an industrial feasible point at this point for any industry to take up that problem and go ahead. Because if an algae can be, that biomass with that much oil can be produced, then that is feasible for an industry to take it up.
Okay. Our another question, Bangladesh audience, that actually she is asking why algae? I mean, instead of other biological sources like 2G, 3G biofuel, why we are moving towards algae? What are the benefit or the advantage of using algae
So,
for this product?
so I'm not telling only algae. We should have multiple sources for multiple things. So you have different kinds of food cuisines, right? You don't
Yes.
have only one particular cuisine. So you have a different kind of the same thing. You should have an alternative. But the problem is, for the advantage with this algae is they are photosynthetic. They does not require much of inputs as a cost process, and they can convert, especially algae can turn up your environmental hazards into a nutritional process later. So that's the main relevance of algae. So the second generation is has its own importance in production of bioethanol, which is very huge, and also some high value products are also being produced. I'm not telling that second generation or third generation, all are expected to meet for different products. Even algae is producing biodiesel, and second generation by sugarcane bagasse is producing bioethanol. So there are two different commodities which are equally important for different things. So biodiesel for algae is a main precursor for biodiesel.
Okay. Another question is like, uh, I mean, in Bangladesh, basically we cannot adopt or support this process for biofuel production
Mm.
of cost that we discussed earlier.
Yeah.
But our biotechnology graduate or other interested students, they actually wanted to know how we can do, I mean, how we can involve in this process for future biofuel production. As right now, like we do not have much GCMS facilities or other facilities. So would you please shed some lights on algal bioinformatics that we can start with right now?
Yes, definitely. To give a glance of that, this has a different sections. Like if you, there is still a lot of things has to be explored in algal biofuels, especially to understand the genomics of the algae is very challenging. So it is a very relevant that because nowadays everybody has a computational power at everywhere, and this is how we are able to also talk to each other even without going anywhere. That's a very good way where we can approach anywhere in the world, right? So bioinformatics is one source where we can contact with each other and we can do it manually. And there are a lot of web tools which are available where we can sort it out at initial level and we can screen it out. For example, you can work on the genomic aspects and you can assemble or annotate those there are number of genes which are available, especially for M.Tech or B.Tech or M.Sc or graduate or postgraduate students. This is a very good project where they can just pull out the scaffolds or genome information which is present, and you can make that information into very important functional annotation that will be useful in due process. Thus, you can use the bioinformatics as a tool. And also, whatever microalgae you are trying to find out in a nature, that can be screened, and there are simple techniques what we do in our lab that can be done and that can be standardized and kept ahead. So you don't require especially GCMS, but the basic studies can be established. And if you can establish a culture collection with all these basic steps, that can be a repository for a future once the government of Bangladesh or any other collaborative institutes or countries are ready to have it, that you can integrate with that and we can have. See, we want to see the whole world as a single entity,
Yes.
because everybody needs a biofuel, everybody needs a high value product. It is not that just it is made in one country and it's not, but everybody. So if you have a consortium of microalgae at a preliminary level, and I think we can have more and more interactive programs, and ICGEB also supports such kind of programs that can benefit even any student who comes from Bangladesh or any part of the world.
Okay, another question is very interesting question. Like algal genes, I mean, those are producing this biofuel. Because downstream processing and other things are very much complicated in algal system to get the
Yeah.
biofuel. So how
Yeah.
about using those genes and I mean, using genetic engineering, can we use alternative host like yeast or others? I mean, for manipulation?
Yes, there are some studies people use oleaginous yeast. They are also just like oleaginous algae. Oleaginous yeast are also present to produce these PAMs or fatty acid methyl esters. As I said, the advantage with algae is you don't require any inputs for that. So it's our natural inputs and a minimal cost inputs. But when you use a yeast or E. coli, you need a lot of glucose or any other sugars which has to be supplemented, and that is a cost factor. So if you are trying to produce an high value product which is having a high cost, then it is feasible. So if you want to, and also mostly the glucose or any hexose sugar which is being used, that is used for production of bioethanol, not for biodiesel or lipid production. So thus there is both the systems have their own privileges in using it, and both have like for example, the fermentation by yeast cannot be done by algae, but the fixation of carbon dioxide from algae cannot be done by yeast. So both have advantages and disadvantages in their own way. That's why I say that each system should be fine-tuned for getting its own relevance.
Okay, and then what is the difference between biodiesel and bioethanol?
Biodiesel is just a component of diesel, which is the major component of that. Bioethanol is a blending with the petroleum. This thing. So we have the 5% blend, 10% blend, pure bioethanol plants which run our automobile. This thing, but the requirement of bioethanol is mainly for the automobile cars and all those things. But biodiesel is a major for transportation. So there is a chemistry difference, and also there is also it has to be blended with petrol. So in Brazil they have 100% bioethanol where they can run the cars. So that is a huge difference. So second generation biofuels are the ones like sugarcane, bagasse, and all. After fermentation, you produce bioethanol that can be used in different forms from in an industry, but biodiesel is the one, the transportation fuel, what we are requiring.
Okay, so during downstream processing, so
Yeah.
this downstream processing, I mean, one single host can produce both, or we have to go for a different way?
No, it's entirely different. It's independent of that. I cannot produce bioethanol with biodiesel. I can produce a high value product with bioethanol. I can produce biodiesel with the high value product, but not with that. There is no combination in both the things.
Okay.
Because the carbon, everything is carbon
Yes.
and nitrogen. So
Yes.
carbon is getting go either to bioethanol. The process of fermentation in yeast is done for bioethanol, and we don't do fermentation for biodiesel. Biodiesel is a lipid which is transesterified, and that can be used. And now there is a process called hydrothermal liquefaction, where you can just take the oil, take the algal cells, put it and heat it to the high temperature, and you get a crude oil which is called crude oil, which can be refined in the biorefinery into different grade oils, just like what we do with the fossil fuels.
Okay, that's great. And then this very important question from Bangladesh Sugar Crop Research Institute. We have a Sugar Crop Research Institute where mainly sugarcane, sugar beet, stevia, and other platform are using for producing the sugar, basically.
Yes.
The derivatives, the question from that Sugarcane Research Institute, if they can produce bioethanol from sugarcane rather than producing sugar, how effective that is?
The simple solution is that is what the Brazil is doing. Brazil is the highest producer of sugarcane, and bioethanol plants. What is being produced in Brazil is from sugarcane bagasse. So the challenge with it is possible. In India also we are establishing a lot of bioethanol plants in HPCL, like Hindustan Petroleum and Indian Oil Corporation, where we can use this different kinds of sugarcane, this thing. And Brazil is the best example for producing bioethanol from sugarcane. It's well known. The only drawback is the enzymes, and there is a monopoly till date now, because only Novozymes is the one company which can give the enzymes for breaking down into cellulose and then later for fermentation. If something can be done on enzymes, and a lot of research in worldwide has been done on enzymes, and even at ICGEB we have developed new enzymes to cut short these sugars and release the celluloses into all these components. So this is a very, very successful phenomena, and it can be done.
So that means biotechnologists can do a lot for producing enzyme first. I mean,
Yes.
they can have work on enzyme production, and
Yeah.
then using those enzymes, converting different substrate to biodiesel or bioethanol.
Yeah, that is the major cost for any of the bioethanol production. If you can produce an indigenous or a native enzyme production in your own thing, then the whole cost comes down. So that is the main reason. So now the monopoly is only with one huge company, but if you have your native enzymes, you can just do that. Otherwise, also the biorefinery companies, bioethanol producing companies are already using the Novozymes, CTEC2, CTEC3 and all. These are very well engineered and well studied in due course of time. Even at ICGEB, our colleagues work on enzyme engineering of these different, we generate different kind of chimeric enzymes for this biodiesel production, bioethanol production.
Okay, so I have a few more questions that participants sent us before.
Mm-hmm.
How successful can biofuel production be without harming agricultural production?
Yeah, yeah, that's already I think I stated in my lecture also. So we should be very clear that we are not competing with food. Food is important thing. Fuel is also equally important, right? So we have to look for the alternative sources. For example, algae. You don't want any. So there are a lot of barren lands, are unused, uncultivated lands, which can be used for growing up the algae. So even you can grow at the coastal belts where it can be done. You can use in eutrophied lakes or waste lakes where you can grow the algae. So there are a lot of possibilities where you are not actually mimicking your food sources. So that's the main thing why the first generation biofuels have been diverted and people walk away to the second generation biofuels. So there is a huge debate because we cannot compromise on food, this thing and food. So equally we have to balance. That's how we have to go ahead.
One technical question: can algal biofuel also be a better option for phytosequestration process?
Okay, sequestration process, yes, definitely. That's where popular now called mission innovation or clean technologies, what we are working upon in different countries, are looking for that sequestration. And algae is the only source where you can sequester and you can produce a lot of oxygen back. That's why it is called carbon neutral or carbon negative. And this is a very promising system that was done in millions of years ago. And that's why we are living on this beautiful earth, and that can be redone with understanding the algal ecosystem.
Do we have any success story rather than research on biofuel production?
Yes, bioethanol we have a few plants which are running on. At this point, algal biofuels, especially with the production of biodiesel, there is not a commercial plant in the world. But people are with bioethanol, there are a lot of plants which are very successful. But as I said, like Brazil produces bioethanol, which can run cars, automobiles, and all those 100% bioethanol.
Okay, thank you so much. Actually, the weather in Bangladesh is getting worse, I think, and then we are here for a long time. I'm requesting Mr. Tanvir Ahmed, if you have any more question.
So Tanbir Ahmed Piash is our alumni and he's a lecturer at Bangabandhu.
Thank you, sir, for your very huge and elaborative presentation, and huge number of participants participated to this session. And sir, Professor Abdullah Mohammed Shoail sir already asked you the most of the question I have noted down, and the most of the question have been answered by yourself, and no more question left to myself. And thank you. I am again I thanking you so much, because lots of participants have praised of this program, because they have come to know as much as they did not desire. Actually, it was a huge presentation and informative presentation. Thank you, sir.
Yeah, definitely.
Okay. Dr. Jutur, I had a question. EU Parliament is calling a complete ban on first generation biofuels. So do you have a say on that? Well, what's your reaction? By 2020, they are calling for a complete ban.
Yes, because that is the first generation biofuels are mainly food crops, edible crops. So that's the reason why people are almost shifted from the first generation biofuel, and we are looking for the alternative sources. So the fourth generation is the most advanced version of second and third, where you can do engineering aspects and all those things of the microalgae or anything to just generate the biofuel. So first generation, because it's all from edible sources, we cannot compromise. That's the main reason they are just shifting. That is actually a reasonable thing, because we have many problems with agriculture, this thing where we have to improve the crop for the whole growing population, and that is actually a feasible solution. We move away because there are a lot of non-edible crops which can be used for biofuel production, because the basic biochemistry or biomolecule structure does not change. That's why we have to understand each compositional level. That will give the answer for us.
Is the fourth generation environment friendly?
Yes, that's the main reason. So we are trying to recycle the environment, and also you producing the biofuel, which is carbon neutral or carbon negative. So that is
that's great.
Exactly the environmental friendly nature. Yeah.
So if we don't have any more questions, then we'll end today's live. So I'll ask Dr. Abdullah Mohammed Shoail, sir. Do you have any more questions, sir?
No, we do not have any more questions. So I would like to thank you, Dr. Pavan Jutur, from core of my heart and from my organization Science Portal BD and Cell Genetics and Plant Biotechnology Laboratory, Jahangirnagar University, and then all of my teams who are working very hard here, and then all of my colleagues. Thank you so much. Thanks all. And then we would like to invite you to like, share, and comments on Science Portal BD to get future update.
And also, I'm inviting Dr. Jutur to come in Bangladesh, come and visit Bangladesh.
Oh, definitely.
You are welcome here.
Sure, definitely. I'll plan definitely. And I will be happy if you have any queries, please email me, and we can be in touch with it. And I'm happy to collaborate with any person who is interested with algal biofuels. And as I said, ICGEB has a huge opportunity of fellowships, research programs for the faculty, students, and undergraduates, graduates. Please try to look for those things, and you can have a glance of that. And have a good day. And once again, Dr. Professor Shoail, thank you for providing this opportunity, and happy to meet you again. And
thank you
goodbye,
so much.
everybody. Thank you.